Hey everyone, welcome to our third genetics discussion video. Today's topic will be the concepts from Mendelian genetics. But first, let's go through some definitions, okay? So phenotype is the physical characteristics of an organism. Genotype is the genetic characteristic of an organism. Then we have genes, which are the basic physical and functional unit of heredity. These genes are made up of DNA, and some of them act as instructions to make molecules called proteins. Alleles are a variant form of a given gene, meaning that it is one of two or more versions of the same of a known mutation at the same place on a chromosome, okay? So next is homozygous. Homozygous refers to a particular gene that has identical alleles on both homologous and homologous chromosomes, whereas heterozygous has two different alleles of a particular gene, okay? A Punnett square is a square diagram that is used to predict the genotypes of a particular cross or breeding experiment, okay? So it is essentially a way to predict genotypes of the offspring in a specific experiment or depending on what you're up to. A test cross is a method used to determine the zygosity of the former by analyzing proportions of offspring phenotypes. So in this case, they involve the breeding of an individual with a phenotypically recessive individual to determine whether it was a homozygous or heterozygous dominant phenotype, okay? So that's what test cross does. Next, we have the Mendelian laws of inheritance. So the first thing that we know is that these alleles, they exist in pairs, right? Because homologous chromosomes, you have one allele from each parent. Then you have the law of segregation. The law of segregation just means that during gamete formation, the alleles for each gene segregate from each other so that each gamete carries only one allele for each gene. So segregation involves the separation of alleles for one specific gene or contrasting elements at the same locus, okay? Next, you have the law of independent assortment. Independent assortment means that genes for different traits can separate out independently during the formation of gametes. Now we have the law of dominance. So in this case, it's just a fact that some alleles are dominant while others are recessive. An organism with at least one dominant allele will display the effect of the dominant allele in the phenotype. So the dominant allele dominates or covers or masks the effect of the recessive allele, okay? If there is at least one of them present. Okay, so how does meiosis help explain these two laws that we have? So segregation describes how homologous chromosomes and hence allele pairs in this case are separated within meiosis 1. Whereas with independent assortment of alleles, that describes how homologous those pairs align randomly during metaphase 1, okay? So that's how meiosis ends up explaining these two different laws that Mendel proposed. So how does independent assortment and genetic recombination during meiosis lead to the enormous diversity that you see between gametes? And this is because that, so remember from last class, recombination or crossing over occurs within prophase 1, where homologous chromosomes pair along each other by length, gene by gene. Sometimes they break, sometimes breaks occur and they rejoin, trading out some of the genes that are involved. The chromosomes now have genes in a unique combination, which leads to more diversity. In the case of independent assortment, that is the process where chromosomes move randomly to the opposite poles. So in a case of a gamete, it will end up with 23 chromosomes after meiosis, which means with independent assortment, that means that each gamete will have one of many different combinations of chromosomes. So that's how that leads to more diversity between gametes and how it goes on into the next generation. So monohybrid crosses, those are, so monohybrid crosses are mating between individuals who have different alleles at one genetic locus of interest. And these show, so again, to see a Punnett square here, that is how you would predict the different genotypes. The probabilities of getting one big P from the male or sperm is a half. From the egg, it's also a half in this case. So that gives you a genotypic frequency of 1/4, okay? Then you have the same thing going on with the recessive allele, with these two being heterozygous, right? So the half and half multiplied genotypic frequency ends up 1/4 in this case, if it's going by a monohybrid cross. Heterozygotes. Next, you have the heterozygous, which means that order doesn't, if order matters, that's how it comes out. So you have a big P from the sperm, little p from the egg. Here you have little p from the sperm, and egg gets another half. So that's how you get these values. And then to get the genotypic ratio probability, you'd have to get 1/2 of it. Okay, so that just adding those two values together. So you see 3/4 purple because there's at least one dominant allele in every flower, whereas there is a quarter that is white, which means the both recessive alleles lead to a white phenotype, okay? So that's monohybrid crosses. Dihybrids end up is a similar thing to monohybrid crosses, except they're looking at two different traits in one go. One is heterozygous, which means that it carries two different alleles at a different, at a particular genetic location or a locus. So this actually shows us that the law of independent assortment actually exists, because the inheritance of one locus has no influence over the inheritance of another locus, okay? And these dihybrid crosses typically show a 9:3 to 3:1 ratio, okay? And it can have modified ratios depending on other factors, but you don't have to cover that just yet in this topic at least. That would be next week's problem. So next we have what is called the binomial theorem. The binomial theorem calculates the probability of alternative ways to achieve combination of events. So the expression for binomial theorem is a plus b all to the power of n equals 1, where n is the number of trials, and a and b are the probabilities of two alternatives outcomes. So this is essentially them all expanded out. You do not have to memorize this, but I've put up a video that can explain it another way where you can derive it without memorizing, okay? So definitely take a look at that video. It'll help a lot sorting out this method, this itself. But for now, we're gonna focus on these guys. Let's do a problem and it'll become more clear. What is the probability in a family of 5 children that 1 will be male and 4 will be female? So we're gonna split it up between A and B, where A is the probability of male equals half, and B equals the probability of female, which is also a half, so 50-50 chance in that case. You can put it either way as long as you be consistent with it. In this case, it wouldn't matter because the probabilities are the same, but make sure you guys are vigilant about which probabilities you're using, okay? So in this case, the value of n would be 5. So you take a plus b to the 5th power and you take that expansion and you'll be like, okay, so we're looking at a family of 5 where you want 1 male, so 1A and 4 Bs, right? So you want A to the power of 1 and B to the power of 4. So which of these guys have this term? This guy, right? So we take that term and we just plug that in. That is your probability for this case, okay? So you put that in, you get this answer. So 5 out of 32 would be the probability that in a family of 5 children, that 1 will be male and the other 4 will be female, okay? So the next thing I want to talk to you about is the chi-square test. I'm only going to go over the concept here, but we'll cover more the questions in the discussion itself. So if you guys have any question regarding problems, definitely look at the video for that. The chi-square test is intended to test how likely it is an observed distribution is due to chance. So this is the formula for chi-square test, which is the summation of the difference between observed and expected squared over the expected value. Okay, so for each category you look at, it's the difference of those two values over the squared over the expected value. And this test allows us to calculate the probability of how much of a chance that there is the deviation of probability of a chance deviation of this magnitude from the expected 1 to 1 to 1 to 1 ratio. Okay. The 4 ones just typically represent the different types of genotypes you can have. Okay. So that's the chi-square test. Next, we're going to look into pedigrees. Pedigrees involve autosomal, so it essentially gives you a history of a family or an individual depending on the scenario you're looking at. So squares are male, circles are female. When they're connected like these are the parents, bringing it down to the children, parents, children, coming down like this, okay? So in this case, we see an autosomal recessive pedigree. It generally appears in the progeny of unaffected parents. Okay, so that's the typical one. This is both recessive, so everything just comes down. It affects both males and females since it's autosomal. Autosomal dominant, the allele that is affecting everything is dominant. The phenotype itself appears in every generation, and the affected parents are the ones transmitting it to their children. And it's equally represented in both sexes, okay? The next one is X-linked recessive. This guy has affected more men than women. No offspring of an affected male shows the phenotype, but his daughters are carriers, okay? So since the allele is recessive, it doesn't show up, but the daughters are all carriers, okay? But then half of the sons of these daughters end up showing that phenotype because it's on the X chromosome and nothing is there to block it off, okay? Because that allele is recessive. So once one of the males get it, because you only get the male, the sons only get an X chromosome from their mothers, so they end up affected more often than the daughters, okay? So next up, we have the X-linked dominant. This is where affected males pass the condition on to all their daughters, but none of their sons. So males to their daughters, but none of the sons, okay? Because it's X-linked. Sons do not get an X chromosome from their father. They only get a Y chromosome. Affected heterozygous females married to unaffected males pass the condition on to half their sons and daughters. Okay? So that is all I have in terms of the concepts here. Definitely do the quiz. Quiz number 3 should be up on Blackboard under Module 1. This is the information in Mendelian genetics. This is the password, so make sure you guys look this up. If you guys have any questions, feel free to let me know. Okay, have a good day.